Okay, I think most people are here. Welcome, everybody. I hope you had a nice traveling to Japan and you're all not too jet-lagged, so you can follow along. As you probably know, since you registered for this workshop, we are going to talk about two different software ecosystems that are now collaborating more and more. So we have now this joint workshop. One of them is GNPS-2, which you can see here on the slide. That will be the first part. And then the second part will be MetaboLights, which is another ecosystem, and they will also introduce themselves when their part will then start, so we'll hear more about them. So the first half will just be about GNPS. You can already see you have some QR code here. If you scan that, that will actually bring you to a Google Doc, and there you will have some material, including all the slides that you will see today, that if you want to have access to them later, you will be able to access those. You will also have a link in there to another Google Doc, and we thought that might be nice. It's actually a public note sheet, so you can take notes on this other slide, and you can also type questions in there if for whatever reason you don't want to raise your hand in the middle of the workshop. It will be actually pretty interactive, so there might be times when you're not sure, oh, I missed something, what should I click next? Then you can just raise your hand, or you can just type in this other Google Doc your question, and then you can see there are always a lot of people here, so we will try to answer in this Google Sheet. Okay? Has everybody scanned this QR code? Who wants to? Okay, at least nobody seems to object too strongly. So one thing we would like to start with is to get a little bit of an overview over where you all actually come from, like what kind of data you've worked with in the past, what kind of knowledge you already have, also that we can maybe adapt a little bit to what you already know. So here, another QR code, please, for you to scan. So it would be cool if you could do that. Then we will go to some just very short survey questions to understand what kind of data you have already worked on. Have you all scanned this code? Because the slide will vanish in some seconds. I still see some phones. Okay, I think everybody has scanned it. Yes, that's good. Okay, so after you did that, you should now see some questions showing up. For that, I will also jump over here to the here. So the first question that we have, where we would like to know where you all stand, is what type of data you actually routinely work with, whether it's NMR, LCMS, GCMS, whether you rather use public services for metabolomics like Metabolome, Bureaucrates, or you maybe don't work with metabolomics data at all because you're just here to learn. So in my case, LCMS. And then we should be able to see here how the answers are accumulating. Oh, okay, a lot of LCMS. That's good, because that's basically everything, at least in the GNPS part we were talking about. A little bit of NMR. Okay. Okay, great. Majority LCMS. Next question. If you have worked with LCMS data, on which samples do you routinely require MS-MS scans? Do you do it for each sample that you inject? Only the QCs maybe? Do you not use MS2 at all and just work with MS1 data? Or, yeah, you don't work with MS data? So in my case, it's all samples. Oh, wow, okay. Okay, so a lot of non-targeted people here, apparently. That's cool. Oh, wow. Okay, let's move to the next one. So since we're talking about public data here today a lot, we would like to know if you actually ever have deposited data in the public space on any of these repositories, like GNPS-MassIVE, MetaboLights, Metabolomics Workbench, Zenodo, any others, or whether you didn't. In my case, it's actually two of these answers, so I will just take this one now. That. Okay. Okay, great. Okay, so most not at all. So we hope that this workshop will change this. It's still moving? Okay, awesome. Do we have one more? No, right? No, perfect. Oh, okay. GNPS is catching up. Okay, great. That gives us a nice overview of what you have all been doing in the past. Let's jump here a bit ahead, since this worked very well. Okay. So some of the things that we are going to talk about today, like I've seen most of you have already worked with LCMS data. That is very good, since we won't really go through the basics anymore. If you still have some questions that you feel like you need answers to really follow along, as I said, you can type questions into this Google Doc if you are, for whatever reason, not willing to ask during the workshop, and then we will try to answer this, but it seemed like most of you are up to date with that. Then MZML files. Does anybody not know what MZML files are? If not, you can still answer. You can still ask now or later, just in the Google Doc, and we can answer that. But this will be the kind of knowledge we basically kind of like build on top of as we go through the flow of the presentation. But you can still ask if you don't know. Also, in GNPS2, you can actually create accounts. We won't actually need an account during this workshop, so you don't need to bother with this. But if you want an account for whatever reason, you can request a token via this link here. And yeah, you don't need to... okay, it's on the next slide actually. Sorry. This link here, we have a QR code actually for it. So if you scan that, you can request a token for which you can sign up to GNPS2. And you can then run all kinds of analysis there. But as I said, you won't need this for this workshop now. This is just if you're interested to actually do this in the future. And yeah, with that, you can see we are quite a lot of people, so we will circle through a little bit. So the first person I will actually hand off to, I don't want to steal any slides, is Helena, and she will start off and tell a little bit about how you can use GNPS.
Hello, everyone. So I am Helena, and well, on the hands-on part of this workshop, we are going to talk about an untargeted metabolomics data set that we acquired in our lab that are fecal samples from felines, from cats, that were acquired in our lab. And well, when I say cats, you're probably thinking about these very cute little creatures that some of you might have as pets at home waiting for you to come back for the conference. But this data set was particularly acquired in samples from the large felines, so lions, tigers, and cheetahs, and so on. So we have a very special collaborator in our lab, Dr. Lee Hagee, that he had all these fecal samples from these large felines from the Project Survival Cat Haven in California. So this is a public data set that is available on GNPS-MassIVE. And the first tool that we are going to use to explore this data set is the GNPS dashboard. Basically, the GNPS dashboard is a LCMS web visualizer. And what can I do with the GNPS dashboard? Many things. So first of all, you can, for instance, inspect public metabolomics data sets that are deposited in the main repositories, such as MetaboLights, Metabolomics Workbench, and GNPS-MassIVE. You can also use the GNPS dashboard to inspect proteomics data sets deposited in PRIDE and MassIVE. And even if you have just an MZML file or a RAW file, you can also just upload that in the web browser and inspect that further too. So if you have also conducted GNPS analysis, for instance, molecular network, and for those who are already familiar with that, you can also directly explore the files that were used to run your task in GNPS directly in the GNPS dashboard. It also allows interactions between collaborators, so it's very easy for you to share exactly what you are seeing in the GNPS dashboard in your computer with the other with collaborators, for instance. And it also allows like synchronous analysis, which is very good for teaching. And it also allows to launch other GNPS analysis as well directly from the GNPS dashboard. I know it can be overwhelming for people that have never used the GNPS dashboard, but the idea here is to give you a brief overview on how you can use it, mainly to explore public data sets that are on the public domain. Here, this left-hand part. So let's go to the hands-on part of this workshop. So for those who want to follow on your computers, I would like you to go to this explorer.gnps2.org. I'm also going to go hands-on with you. Oops. I'm going to give you like some seconds. So it's here, like explorer.gnps2.org. And then here, what we will do is put the dataset ID that will have all those fecal samples that I previously explained to you. So this is the dataset ID that we want to search for. So on the very first part here, on the first field in this page, we are going to type MSV 0000 93073. It's probably taking a while because there are many people doing that, so let's wait a little bit. So while it's loading, I would also like to emphasize that, like here, we are putting a dataset ID of a dataset that is deposited on GNPS-MassIVE. But if, like, you have a particular dataset in MetaboLights or Metabolomics Workbench, you can also use the dataset ID of these other repositories to put here. And basically, this web page is going to give us like an overview of that dataset. It's still loading, but it's going to give us like the dataset title and the dataset description as well, and a table with all the raw files, with all the files that are deposited as part of that dataset. It's down. Oh no, like for you it also didn't. Well, that can happen in workshops when there are many people trying to use at the same time. But, well, I have the backup slides here, so maybe you can follow that later on. I'm going to leave it here, like trying to load for a little bit. So, well, I'm going back to the slides. But if any of you like see that it worked for you, like if finished updating this this tab, let me know. And I perhaps can come back here. It worked. Okay, let me try. Oh, yeah, it also finished for me. You see, like it was loading before, but now you can see here this blue square that shows, okay, that the dataset was successfully loaded. And the title here, feline fecal samples, Orbitrap MS data. And then we also see the description here, fecal samples from 14 different species of felines, and like the extraction method used and so on. So if we scroll down here, we will see a table that will have all the files that are deposited as part of this data set. So let's say, for instance, here the very first one is the extraction blank. We don't want to inspect that now. But so like let's focus on the second row here, like this A1QE2.mzml file. So for us to select that, we can click on the left square here. And then we can go down, and we're going to see these blue buttons. And then we can click here on visualize one file in GNPS2 dashboard. So you can click here. It'll take a little bit. Yeah. Okay. So here, the very first section that we have here on the left side is the file selection. So here we see this what we call like GNPS USI. It's basically like USI stands for Universal Spectrum Identifier. It's basically a combination of strings that is going to tell exactly in like what is the data set that a particular file is coming from. So here we can see, okay, it's a mass spec data that comes from this particular data set that is in this particular folder, and it's this particular MZML file that we are inspecting here on the web interface. So it was loaded successfully. And we can go down, and here we are going to have like some viewer options. I'm not going to play with that right now, but like what is important to see is like there are these ready green signs here. It means that the file was successfully loaded into the GNPS dashboard, and we can do like some visualizations and downstream analysis with that. Is it working, the dashboard, with you? Like for people that are following? Okay, great, great. Okay, so here, if we scroll down, we will see this data exploration section. So here is an overview of your analysis. On the x-axis, we have the retention time of the analysis, and on the y-axis, we have the M over Z. So this was acquired in positive mode, okay? And here on this heat map, we can see like the abundances of the MS1s that were collected, and all these blue crosses, they show all the MS2s that were triggered in the analysis. And if we scroll down again, we will see already the TIC plot. So here's the chromatogram. Okay, so we have already successfully loaded this file in the GNPS dashboard. But now we want to say, okay, I want to explore like a particular compound in this file. How can I do that? So I'm going to go quick back to my presentation. By the way, like all of the step-by-steps are here on the slide, so if you like want to follow it again later on, you'll have all the step-by-steps. But well, I'm coming back here to this presentation. So there is an interesting class of compounds that our lab has been investigating for a while now. It's this like microbially conjugated bile acids. So there was a study that was conducted in our lab that discovered these bile acids that can be conjugated with other amino acids. So, and what was very interesting to see is that these compounds, they are only found in the colonized mice, but they were completely absent in the germ-free group. So, well, then I want to see, okay, is this compound present in the fecal samples from the felines? So we have already here the mass to charge ratio that we are interested in. So this would be relative to like a protonated molecule. So I'm going to copy here, because I want to extract that ion from the chromatogram. Oh, sorry, not here. Here. So if we scroll all the way up, we're going to see the XIC options, and then we can enter the mass to charge ratio here in this field. So I'm just copying paste. So it's 506.3840. So we're basically saying that we are interested in extracting that ion, and then we need to set up the tolerances, right? So since this is Orbitrap data, we can put, for instance, 10 ppm, it's fine. It's already pre-populated here as 10 ppm. But then we must change here the tolerance unit from Dalton to ppm. So we scroll down, and then here on the left side, we are going to see the extracted ion chromatogram. And all these red crosses are showing all the MS2s that were triggered for that particular precursor ion. And then we can, for instance, click here on the apex of the peak. And then right away, we already have the MS2 of that ion, of that particular mass to charge ratio. And I mean, isn't that cool? Because, like, let's say that you were going to do that in the traditional way, like, oh, I want to explore a data set that is in MetaboLights or GNPS or Metabolomics Workbench. You will have to go to the website and then download the file, and then you would have to perhaps like install a software in your computer and then open it, and then like extract and go to the MS2s. But here you don't need to download anything. Like you already have that information right away. So like if you read like a cool paper that the data set is publicly available in one of these repositories, you can like just go there in the dashboard and explore right away the raw data. So here, I'm almost at the end here for the GNPS dashboard. Here, if we click on View Metabolomics USI, it's going to open a pop-up window, and then here you're going to have your like the MS spectrum that you have just selected, and if you want, you can download all this information as a text with all the intensities and all the M over Z's here. You can download it as a CSV file or as a JSON if you'd like. And if, for instance, you want like a high-resolution figure to put in a report or like in a manuscript, you can also download as PNG or SVG. And I would also like to highlight here that this other like combination of strings is like the USI again. But here, like in addition to having only the file name information, we are also going to have the scan here. So this is the scan for 2,471. So with this USI, with this combination of strings, we are already saying, okay, it's this data set, this file, and this particular scan in that file. And with that, we can do many other downstream analysis that Simone is going to take over now. So.
Okay, hi everyone. My name is Simone. I'm also a postdoc in the Dorrestein lab. So we arrive at this point, right? We have a scan that you are very interested in. And here now we are going to show the power of doing public data repository reanalysis. So usually, I don't know what kind of metabolomics you guys are doing, but usually we have this big problem in the untargeted world where the metabolites of interest are usually not annotated. So we have no idea of what they are. So the idea that we had is that even if we cannot tell you what is this molecule, maybe we can tell you where this molecule was already observed. And that could help you a lot for the biology. So in this case, Helena already told you that the molecule that we are investigating is a new microbial metabolite, but maybe you don't know this, right? Maybe you're just working with an unknown molecule. And the question is like, is this of microbial origin? So I'll just introduce you to one of the domain masts that we have recently created that is called MicroMast, that helps you basically leverage all the data that has been already deposited in GNPS-MassIVE and soon also in the one in MetaboLights and Metabolomics Workbench to search specifically your MS spectrum against the billions of spectra that have already been deposited online. So MAST basically is a search tool. In this case, MicroMast, as the name says, focuses specifically only on molecules that have been acquired from microbes. And for microbes, we are talking about bacteria, fungi, archaea, or like other very small organisms. And what we did to build MicroMast basically is just to restrict and create a specific repository of all the SMS spectra that were exclusively acquired from monocultures of bacteria. With the idea is that if the spectrum is found in this kind of sample, it's probably of microbial origin. Obviously, I cannot go really into the details, but we have put a lot of safeguards in places about how to filter for like medium blank controls and other contaminants. The paper was recently published, so if you want to know more about MicroMast, you can find it online, and then you can also get in touch with us and we will be able to go through your questions. So let's go to the hands-on part of MicroMast. And so if you guys can please go through that link, you can just click on it. If you are following on the PowerPoint, you can just write MicroMast on Google. Make sure that is must.gnps2 in the URL. So I'll just give you a second. I have already opened it. That is here. So we should all be on this page. So what we are going to do next, basically, it's just representing the case scenario where you are looking for your spectrum of interest, and in this case is the spectrum that Helena previously described. So you should already have the USI. If you don't have the USI open somewhere or available, you can find it in slide 29. It's here. You just have to copy paste it and put it in MicroMust. So you have to just copy paste it here where it says spectrum USI. So while you guys are pasting it or writing it down, as you prefer, I'll just quickly give you an introduction of the web page of MicroMust. So this tool that allows you to search for MSMS spectra against the defined repository is available as a web interface or also as a Python code that also allows you to do batch searches. But in this case, we'll just focus on one single search for one single MSMS spectrum. You can see here that we have two different tabs. One is Spectrum USI and one is Spectrum Peaks. What you can do is just search a USI, or if you are more traditional and you don't have a USI, you can also just copy-paste your fragments, ion, and their intensity and put a precursor mass here. We already have defined like search tolerance parameters, like the parent mass tolerance, the fragment tolerance, and the different cosine score threshold that we are going to use, and the number of minimum matching peaks that we want to use. For the one that are not familiar with cosine score similarity, is basically a metric that we use for molecular networking also, that just compares to MSMS spectra and tells you how much similar they are to each other. With a value of 0.7 above, we have kind of a good we are quite confident that they can be the same molecule or like a molecule that is strictly associated with the one that you have searched for. So if you manage to put the USI in Spectrum USI, what you have to do next is just click MicroMust, search by MicroMust by USI. So I just want to highlight that if you are using the USI, you have to click search MicroMust USI. If instead you have put like the spectrum peaks and their fragments, and you have to click search MicroMust by spectrum peaks. It should be quite intuitive in this case. Another thing that I want to highlight is that when you are doing this kind of analysis, and this also works for the GNPS dashboard, you can always share links to your collaborators. So let's say we are here in Osaka now, and I'm searching for this specific spectrum that I'm interested in. I can just copy the link and send it to my collaborator back in San Diego, and they will just be able to open the same exact results without having you to send the data, download softwares, be on the phone and try to understand which scan you're talking about. And this is, again, the power of like public tools and public data when it's deposited online and everyone can get access to it. So once you have searched for MicroMust USI, you should be able to obtain this interactive taxonomic tree that, by the way, you can also download by clicking through download. But the tree, it's, as you can see, you can zoom in as far as you want, but the tree is interactive and it shows you basically where your MSMS spectrum of interest was also found. So as I told you before, we have restricted this data repository of microbes only from monoculture of bacteria. So, for example, if you put your cursors on one of the pie charts that they show how many matches were found in the total number of samples that we have for that specific taxon, you can see if I move my cursors on Bifidobacterium, you can see the name, the scientific name, the NCBI IDs that we used our taxonomy tree is based on it, the rank of the taxon, that is genus level, and in this case we can show that we have 2 matching samples within the 227 sample that have been deposited under the genus Bifidobacterium. And then obviously you can see the occurrence rate. In this case also the tree is interactive, so you can collapse it at different levels. So let's say if you want to only show family level, you don't have only to click, but you can also just like collapse it at different level using the show levels function. You can recenter it. And in this case, as you can see, we can show that in the Corybacterialis we have more prevalence for the number of samples that we have that have been acquired in the public domain. 2 out of the 19 samples presented, while for the Bifidobacterialis are way less. So this is the tree that you can play with it. It's kind of figures ready for publication, and you can visualize also, for example, one specific lineage if you're just interested in only one. I didn't show it here. We don't have any matches, luckily for us, but the tree contains also blanks and negative controls that we use also, for example, for human cell lines, if you are only interested to define molecules that are found only in bacteria and not in the host. So there are a bunch of like different negative control that are in place that you can also, that will pop up immediately, and you will say like, oh, my spectrum is found in a lot of bacteria, but also in a lot of blanks or in a lot of like human cell lines, so maybe it's actually not a microbial metabolites, but it's something going on. There is a contaminant. What you can also do is open these 3 different tabs. So we have 3 tabs here at the top, if you can see my cursor. Is the library matches, the dataset matches, and the taxa matches. And also the parameters tab. Here it basically is just recapitulating what parameter you have used for this analysis. So if you click the library matches tab, it will pop up this table. So at the same time, while we are doing the search of this MSMS spectrum against all the repository data that has been acquired on bacteria, we are doing also the search against the GNPS library. I know that we haven't talked about a lot of GNPS environment, but we have a massive public library of molecules, more than 600,000 spectra acquired from standards, or that people have also deposited online. So while we are searching against all the bacteria, we are also searching the spectrum against our library. So hopefully we are able to annotate it for you. If we were not able to find any matches, this tab will be empty. But in this case, we can see that the matching, the spectrum is actually to the leucine or isoleucine, because we cannot define it via MSMS spectrum, conjugated to deoxycholic acid. So this is obviously makes sense because we literally search for the spectrum. So this is kind of cheating. But in your case, if you had no idea what molecules you were looking for, hopefully we would also get an annotation. And here you can see that there is not only one annotation. We provide all the kind of parameters that you can see how well is matching to the library spectrum that we have. So we have a cosine score that, as I was mentioning before, is a measure of the similarity of 2 spectra, and the number of matching peaks, the adducts, the names, and then you can also open the spectrum resolver. So we will move to the next tab, that is the datasets match. So in this case, we will show you to which scan in that specific sample of the bacteria that was analyzed, your MSMS spectrum was found to match. So you can see here that the spectrum was matching a sample from Escherichia coli. Again, you see the NCBI, the rank of the taxa, the cosine score, the number of matching peaks. But what you can also do, it's open the spectrum resolver. So sometimes, if you don't trust cosine score and you are more traditional, you want to probably inspect the molecules. So this is another tool that we offer in the GNPS environment that basically allows you to just check spectra against each other. So if you trust more yourself than bioinformatics tool, here you have it. So you can see here that we have a cosine score of 0.77. You can see the matching peaks I will not go through this, but you can also change the size, the visualization of the spectra, and download SVG files. So also this one will be quite easy for your publication to use. So going back to microbust, and this again, it shows for all the samples that we're matching, you can see them all. You can also show more matches, right? So you can see here there are 20 matches to all the different bacteria, and you can see all the different scans that were found to match. Also in this case, we provide the MassIVE ID. That is something like useful because we have limited metadata associated with the sample, right? Metadata is something very difficult to work with. So we don't include all kind of metadata associated to the sample, but let's say that only specific bifidobacteria were found to match your specific spectrum of interest. What you can do is just click on the MSV datasets. It will open the dataset that was acquired, and here you can find, this doesn't have like a lot of description actually, but you will find a description of which analytical parameters were used maybe to acquire the data, what condition the bacteria was culturing, but most important, you will find out also who deposited the data, so you can contact them, maybe you want to ask them if they have the strain of interest for your research. So finally, we have the taxa matches, and this is kind of just recapitulating all the matches to the different taxonomic level. So obviously, you can see that root is the one that has the highest amount of matches because it comprises all the matches that were found in all the samples that have been deposited in the microbust repository. So you can see that we have 60,000 samples and 45,000 of those were found in bacteria. So the repository is quite big and quite diverse. We have 60,000 samples. This is comprising like more than 100 million MSMS spectra. So there is a lot there. Is it enough? No. But we are open for collaboration. So there will be various iterations of micromass. So if you're acquiring microbial MSMS data through LC, you can deposit your data, and this will also be incorporated in the future version. So hopefully this will just keep growing, and it will become more and more useful to the community. And again, hopefully we are promoting public data reuse. And you can see here, again, all the different levels that you are interested in, and maybe is matching or is helping you to define your a more defined scientific question. All these data sets, all these tables are also downloadable. You can click by either download it as a XLS file or CSV file. So the tree, maybe I can show you this. The tree is quite big, right? Like if you want to visualize it, you are also able to see it all. Well, you cannot see anything there because there is not much, but you can also, if you want to just show maybe, this molecule was not found in any of the other bacteria that we analyzed, you are also able to see that. The tree, it's massive, so it's going to be fun for you to explore it if you're interested. All the data that has been deposited and used for micromass, there is an easy way to access it. So if you go to the paper itself, there will be links to GitHub pages where we have like in tabular form all the information that we have. So how many bifidos, how many bacteria, what table we used, how we build the taxonomic tree. So everything is like public and open, so you can just go there and download and even modify the code if you want. So with that, if we don't have any question, we are basically almost at the end, but feel free to interrupt me if you want to ask anything. We welcome questions. And again, if you are not familiar with any of this tool, and this workshop maybe felt like overwhelming and maybe a bit difficult to follow, we have all the basis for the theory behind our tools, and that we build and we are building, and we are also building the documentation. So the PowerPoint has links to all of this. So you can go there and check the documentation. We are also very responsive via email. So if you want to ask more questions, collaborate with us, you are more than welcome. And what we want to really push also is that please upload your data online. We have seen from the previous question that half of you is not doing it. I'm not going to judge you, but you should do it. So here you have like a direct link on how to upload your data online. So we will have 10 minutes maybe for question. Before going through the question, I will just go through a self-promotion of ourselves. So from the Dorrestein Lab, we are 3 here at the conference. Come chat with us, we don't bite. We also have 2 talks and a poster. So here there is all information. So if you want to come to our session to see our talks and chat more, you will find us here. And since Pieter is not here today, but he will be here tomorrow, and he's always promoting us, we're gonna give him a shout out. So tomorrow he will be obviously presenting at the plenary session. So, and he will talk a lot about big data reuse and how it's very important to deposit public data. And with this, we are at the end of the workshop itself. So if you have any question, please let us know. Any question? Sorry, can you, yes. What?
Yes, it's working now.
Yes.
Can I search for a metabolite in the database, for instance, indolactic acid, and then I just find the spectra and bacteria and everything?
So you can. So what you can do is go into the GNPS library website. So we have a website that is specifically for the library. So all the molecules and all the spectra that are in our libraries are there. So what you can do there, you can search by name of your compound, you can search the SMILE, you can search the chemical formula. There you will also see the information of like who deposited, when was it deposited, was it acquired with QTOF or BTRAP, you will have a lot of information. So what I can suggest you is that you can, if you are interested in indole propionate or other indoles derived microbial molecule, you go and you take all the different library spectra and then you search them all. You can search manually one by one, or you can search them all together through a batch search. In that case, you will have to have more computational skills. But if you have them, come chat with us. I'll describe you how to do that. And that's also is a good question because allows me to highlight another good things that we have in the GNPS libraries is that we have more than 600,000 molecules, right? But there is a lot of like overlapping. So for example, for indole 3 propionate, we have like maybe 5-6 spectra because these spectra are deposited by the community also. So there would be acquired via Orbitrap, with a QTOF, different collision energy. It really depends. So the spectrum, the MassIVE spectrum will change a lot, right? But the fact that we have all these redundancies also help us to help you because if you are just working with QTOF data, maybe your MSMS spectrum will have higher chances to match a spectrum that was acquired with the same instrument.
Great, thank you.
Thank you.
And I have put you the link to the GNPS library into the note sheet, so you can just click on it.
Yeah, I'll show you. So. Yes, this is what I was describing. So this is the GNPS library page. As I was telling you, you can search by SMILE, or otherwise you can search for compound name. Let's say that we want to search for, let's say, in the phenylalanine. Okay, I don't know. This is like a Japanese keyboard. I cannot do a dash. Okay, well, anyway, here you can, let's say, search. I wanted to search phenylalanine acid that we have like very famous as phi-CA, and you will be able to see all the data that was deposited. But as I was mentioning before, you have a lot of information here, like molecular formula, instrument, ionization mode, like Yasin knows better than me, but 90%, I think, or 95% of the data on GNPS-MassIVE is in positive mode. So we mostly work on positive mode. So if you work on with negative mode, it's going to be a bit more complicated. But yeah, you have all the information, and you just have to go here. So you have the spectrum ID, and this is just the little code, the little ID that you need to use, to copy, and just search it. So it should be quite straightforward in that sense. We had a question now.
I want to ask, like in dashboard, you have the TIC view, right? So for instance, if I have 2 more or maybe 3, and I want to compare between the symbol, how can I see it like in TIC with a separate chromatogram?
Can we?
Sorry, I'm not sure if I understood completely. Are you asking how you can see the extracted ion chromatograms in the dashboard?
No, it's like TIC chromogram.
At the TIC, you can see it under the heat map, the TIC chromatogram.
Yeah, I mean, like I want to compare 2 samples at the same time with the TIC.
Oh, I see. I don't think at the moment you can put 2 TICs. I think you would have to open 2 tabs in order to do that. I don't think you can overlay them.
Actually, you can overlay them there, but I mean, like, we can separate them or not.
I don't think we can separate.
But you can separate the TICs?
Yeah.
I mean, it's from 2 different samples, the 2 TICs, right?
Yes, right.
And you want to see if you can separate them in the sense whether they're perfectly overlaying or whether they're shifted? Or?
Yeah. I mean like that, because you guys have the 2, like, and I put the 2 IC inside at the same time, and then they overlap together. I mean, like, may I like just separate them, or?
I see, yeah. At the moment you cannot overlay it now. You could trick a little bit by doing an extracted ion chromatogram with a really big tolerance. Like if you center it kind of, like in the center of the mass range, and then you put a tolerance of like plus minus 500 m/c, then you would get something like that, and there you can overlay it. I can show you maybe after that, because I will quickly try if it works, and if it works, I will show you later how it works, okay?
It's the other way around. She has it overlaid, and she wants to have separate tabs, right?
Yes, right.
It's the other way around, because you can see them overlaid, but you cannot have, let's say, one above the other. That's her question, if I got it right.
Of the extracted or the TIC?
The TIC. But yeah, I think it's not possible as you.
No, I think we don't have it. But yeah, you could like open 2 tabs, screenshotting them and making one on top of each other. But maybe we can tell Bing to add this functionality. This is always a good suggestion. Sometimes you don't want to see them overlaying. Thank you.
Hi. Do you have any recommendations for large data sets? Because I tried once with GNPS dashboard and it kind of crashed. So you have any recommendations on how to make it if I have really big data set, it's just how can I make it work?
Let me rephrase it. So your question is like you want to visualize, let's say, 100 sample or 1,000 sample at the same time in the GNPS dashboard. Yeah. That's something that we don't usually do.
No, not 100, but sometimes like my data sets are quite heavy, so
okay.
A lot of MS2, but
yeah, it's not really meant for that. The dashboard, it's really just like having a quick look at public data without downloading it. If you really want to like do large scale reanalysis, then you should download it, because extracting all of these like files together, hundreds, for that you need a lot of resources, and if too many people do that, then everything will just crash. So it's really just meant for a few samples, having a quick look whether things are co-alluding and look as they expect, or whether a certain spectrum is present without having to go through all of this effort of finding how to download each specific file and then doing all this effort to find out you actually are not interested because it's not what you expected. So yeah, that's what it's for.
Okay,
I hope this answers.
Yeah.
Okay.
Thank you.
Yes?
Yes, I have a question. So we can do metabolite identification over there. So which metabolites, which metabolite database has been used, or how many metabolites have been included in the database?
We have, I think, around 20,000 unique metabolites in the GNPS library that there is redundancy, so that's the 600,000 number, if I'm correct.
Yeah, I think it could be going towards 20,000 metabolites at this point. Not all of them have also smiles. Spectra, it's way more. Spectra, we go into the 100,000s, but unique metabolites, it's like something like 20,000 at the moment. But this will actually increase a lot over the next months because we currently have an effort where we are synthesizing, like it's planned to synthesize like millions of metabolites and put them all on GNPS. So this will like grow more and more in the next months and years. That's the plan. Does this answer the question?
Maybe how many of those metabolites are based on the chemical standards? Or you obtain the metabolite from a public database, or
it's a community resource, right? So every one of you, like a lot of people out of the community, are just contributing spectra to us, and these spectra are the ones that you then see in the library that Simone before called up. So all the spectra you see here in the list, these are community spectra. So somebody in the Metabolomics community provided that spectra to GNPS and said, hey, I have here the spectrum, it's phenylalanine, and now it's here in the GNPS library, and now we have it in all these workflows, and everybody can annotate the data with that. Right? So it's not that we source that directly. I mean, it's also sourced because we try to include things from mass bank and so on, but it's also just people uploading the natural products or we synthesizing stuff and then uploading it there, but it's a mix. So it's always something you should be aware of if you use it, that this is a mix of different things. Some people might have used standards, some people might have used something else, but yeah.
Okay.
And you will be able to see it also here. Usually the libraries have a name.
Okay.
So some of them are coming from matching to the NIST libraries. Some of them are coming from people synthesizing thousands of molecules. This is a good example, for example.
Okay.
This was synthesized actually in our lab. I'm giving this example because we are unbiased. I know more people in our lab. But Emily Gentry, that was a previously postdoc in our group, synthesized thousands of novel bile acids. So she did real chemistry. And then she threw everything in the mass spec. So we have she had retention time. She had they were all separated. And then she acquired a spectra and then took the spectra and put it and generate a library spectrum. So that's, for example, was generated. And you can do it for your own molecules too. For example, I don't know, you are interested in the indoles and you are synthesizing them, you are synthesizing new molecule, you run them through your MSMS, you generate your spectrum specifically, and you can create also a library that can become part of the public domain.
Okay, besides, do we have, or do you provide any identification level for those metabolites identification?
So in this case, the identification level for your experiment itself, because anyway, even if it's a level 1, because Emily knew that she synthesized that molecule, so she knows what MSMS to expect and the parent mass, the retention times. When you are doing your own metabolomics experiment, right, you are doing the annotation through GNPS-MassIVE. That will be an annotation of level 2 because you are matching MSMS to MSMS. So when you are, in your case, in your experiment, you will have a level 2 annotation because you are matching MSMS. You are not running the standard yourself, right? And you don't know the retention time matching. For the library compounds that we have in the libraries, most of them I would assume that could be a level 1 because they are specific standards that have been synthetically created.
Okay. Thank you.
Thank you.
Okay, for the interest of time, we have a 5-minute break now, so if you have any questions, feel free to grab the guys at the front. Before that, Simone, could you go to the Google Doc?
Yes.
We asked you if you could now create an account on MetaboLights Labs. It's not for one of the exercises, you might need it. So yeah, if you could do that in the next 5 minutes, and of course, continue asking questions if you want offline. Great, thanks.
Okay, while we are in this break, I also would like to highlight that, who here works with microbial metabolites, like with microbial metabolites data? who work with plants? There are some. Okay. All the workflow that Simone showed you for microbe mass is also available for plant mass. It's exactly the same thing. But if you are interested in exploring, for instance, in which plants a particular metabolite was found, there is also another website for that. And I'm going to put that in the public notes. So if you want to also use that, it's exactly the same workflow, exactly the same things that Simone showed you.
Oh, yes.
So if you open the...
This one, right?
Yeah, is that one? Yeah. That's it.
But feel free to come and ask questions still if you want. Okay. Yes. So the second part is for metabolites and metabolites data and workflows. Very much here, again, just to reiterate and recap, the objective of this workshop is to make you guys comfortable to consume and reuse, or start to consume and reuse public metabolomics data. I won't spend too much long on this slide, but just to reiterate, data sharing is great. It greatly accelerates the research across the life sciences. It increases scientific reproducibility, reliability, traceability. It enables scientists to quickly react and build upon existing research. It encourages collaborations, increases visibility of your research and the impact of your research, and often nowadays is a requirement for grants and publications. Metabolites. So metabolites can be found at www.ebi.ac.uk/metabolites. It's open sourced and comprised of 2 layers. It's a study repository, which enables the metabolomics community to share findings, protocols, and data. And the second one is it's a metabolite knowledge base or a compound library of information to reference material that is also highly coupled to the database CEBI, which is the chemical entities of biological interest. The database captures information across the entire workflow, so from sample collection through to sample preparation, through to data acquisition, and through to data interpretation. It's cross-species, be that bacteria, plant, or animal samples. It's cross-technique, NMR or mass spectrometry. And it's open access and discoverable through various portals. So the database was developed and is still being developed in conjunction with the metabolomics community. And we aim to create a resource which is very much aligned to the community standards. So, the Metabolomics Society, the Proteomics Standards Initiative, MQAC, the Metabolomics Quality Assurance and Quality Control Consortium. And really the primary requirement for metabolites, for metabolite submission is the raw data. We require the raw data. But we also require it to be supported with rich metadata. So this is how you describe the samples you're using, the protocols. And often we ask you to, metabolites will then underpin this with control vocabularies and ontologies and unique identifiers. And together this represents many of the attributes of the good, fair principles. So make sure that your data is findable, it's accessible, it's interoperable, and then it's reusable. What does a metabolite study look like? Oh, here it is. When you hit the landing page, you'll find information that tells you when the study was public, the release date of it. You'll then get a title related to the abstract, and also then you'll see the publication. So it's easy to find data set. It's easy for a data set and publication link. Underneath that, you'll see a series of tabs that will take you through the actual contents of the study. So you'll have descriptors, you'll have protocols, you'll have samples, assays, metabolites, and then finally the files. Everything is free to download. Often these templates can simply be downloaded, open up in Excel, and exploited. Metabolites does conform to a metadata standard known as the ISA, which is the investigation, study, and assay framework. This is a framework really to standardize the description of complex experimental design. At Metabolites, we provide added value, so we link everything to ontologies. Why use ontologies? Well, it gives semantics to the data, so you can start to visualize, interpret data better, and you get a definition in a hierarchy. So we use ontologies extensively. We use ontologies across species, organisms, and organism parts. We use ontologies across your sample descriptors, your characteristics and factor values. And we use ontologies for instrumentation and also for metabolites. Submissions, Metabolites sees submissions from across the world. The top ones are China, USA, UK, Germany, France. Submissions are increasing for you guys to reuse. We have submissions increasing across all the model organisms, from Homo sapiens to mice to rats and to lots of the plants. And often now what you see is that the data sets are not just published in Metabolomics specific journals, but also in the broader journals, such as Nature and PLOS. The user base for Metabolites is also global, so it's across not just industry, not just academia, but also industry, healthcare providers, and so on. The public data, when you actually start to reuse it, we have about 88% is mass spectrometry data, and the rest is nuclear magnetic resonance spectroscopy data. Most of you guys out in the community are doing untargeted. There's a bit of untargeted and untargeted together. And most of you guys are doing LCMS mass spectrometry. So what we suggest is when you start to consume metabolites data, you can search for the studies on the website. And we have a new test URL for you guys to search. So if you have feedback for the test URL, how better to search all the public data sets out there, we have close over 1,500 now public data sets for you to search. So try the test URL, find us in the conference, and give us feedback. You can also browse the studies online through the FTP and download through the FTP. We also have extensive documentation to help you go through FileZilla, Aspera, Globus, and now we'll show you what we've been working on recently, which is through Galaxy. So I'm almost done with the lecture, and then we're going to pass you to Callum for the hands-on. So at MetaboLights, we have been working on a grant to standardize metabolite annotation workflows for enhancing biological interpretation in metabolomics data repositories. So this means we have to start using the data ourselves. So in collaboration with the University of Liverpool, Imperial College London, the University of Birmingham, and of course us at EBI, we have started to develop standardized workflows to run across all public data sets at the repository scale. And this is to allow you guys to have a standardized output that you can then start to explore. You can start to concatenate studies, you can start to answer different questions, you can start to use different protocols and methods. So we've done this using Metabolites Labs. This can be found at metaboliteslabs.org. And it's very much a Galaxy framework as well. So whatever you can do it, so whatever you see on Metabolites Labs, you'll see at the other Galaxy instances across the world, in Australia, in the European Union, and in America. So I mentioned we built these standardized workflows. So the first one we went for was an LCMS workflow using XCMS. Rick Dunn at the University of Liverpool helped us optimize this process, and now we have it running over all the LCMS data sets at MetaboLights. And we'll show you it's not just restricted to metabolites, but we show you how you can then start to use data from the other repositories, such as Metabolomic Workbench, GNPS-MassIVE, and MetaboBank. One thing we had to start to do was to get data into the Galaxy, so we produced a new data provider workflow. So now we've indexed for you guys to search, and we'll be using this in the hands-on demonstration later, for you guys to work with close to 800 studies now. You'll understand that there's over 1,700 assay files, so these are unique experiments that the public have done and willing to share with you, and indexed close to 250,000 data files for you now to use. The MD data provider tool is basically to take all the data on metabolites and create an open standard file for you to then use with, to couple with all the workflows available for pre-processing. So this will be one of the exercises we'll take you through. So part of this, now we can start to count files. We have close to 60,000, which are Homo sapien data files out there in the public domain. Next one, the plant species, and then so on. We have different organism parts as well, such as leaf, biosample, blood serum, and blood plasma, and we have all the different file formats as well. So yeah, now you can start to couple the MDP tool with with an XCMS workflow to get the outside results. And of course, you can also couple the workflow with my data. So I can bring my data in big scale. I can, at MetaboLights, our public studies are over hundreds of samples. If you want to start to bring your own data, which is hundreds of samples, we can cope with that at MetaboLights Labs. So now I'm going to hand you over to Callum to go through the exercise. We encourage questions during the session, and I'll be around to help anyone who gets lost as well.
Okay, hello everyone. Good afternoon. Konnichiwa. My name is Callum, I'm a colleague of Tom's. I'm going to be taking you through 2 practical exercises to familiarise you both with Galaxy, our specific instance of it, MetaboLights Labs, and browsing MetaboLights studies, finding out how to get information out of them and how to use that information in our tools. Okay. So first things first, if you can navigate to MetaboLights Labs, hopefully you've already created an account. If you haven't, take the time to do so now. At metabolightslabs.org. I'll give everyone a second. So just as a very basic exercise, we're just going to quickly search for a tool just to show you how to browse Galaxy. So I'm going to switch to MetaboLights Labs. Okay, hopefully you all see something that looks like that. Hopefully you've all got MetaboLights Galaxy. So just as an incredibly basic exercise, for instance, Tom was just talking about XCMS. Search for XCMS. And then you can bring up any of the search results. You don't need to understand what you're looking at just now. This is just to introduce you to what it looks like when you're looking for a tool, how the tool prompts you for inputs, and what the various input fields look like. You don't need to understand too much what's going on at the moment. We'll cover tool inputs for a tool in the next exercise. Okay, while everyone's getting on MetaboLights Labs and clicking around, I'll go back to the slides. So the next thing you might want to do while you're on MetaboLights Labs is upload data from a local machine. So you don't necessarily need to do this, but just to show you how to do it, and you can upload data if you like. I'll show you back in MetaboLights Labs. You can click upload data here. This button is accessible always when you're on Galaxy, so it's very easy. You go in here, you choose your local files. This is all in Japanese, so I'll come back to this. But for those of you who can read in Japanese, or you don't have it in Japanese, there we go. If I go into downloads, there we go, click start. There, and you'll see that when you upload anything, it will go into your history on the right. So history on Galaxy is just a record in a session of everything you've done, all the data you've uploaded, all the workflows and all the tools that you've used and the outputs from them. We'll cover this a bit more in a minute, but you can see here that the text file that I've uploaded is in there. Feel free to upload anything if you want, but you don't need to. And I'll very quickly show you, let's just say you don't have any data on your machine, or you tend to keep it in a remote location. You can also use FTP. So if you want to try this out yourself, there's a link just here. This TinyRail link here links to a spectral data file on MetaboLights that you can transfer in. So it's that TinyRail link there. If anyone wants to copy it, or if anyone has the slides to hand, or they want to copy it from the presentation, I'll give them a minute. Got that. So I'm going to copy it myself. Okay, and then from there you can click Paste Fetch Data. And there you can place the link. Okay, and then click Start. Okay, so the helpfully named unnamed data set is being pulled from the EBI FTP remote server. And if I click in here, hopefully I'll see something. It's just the link. Well, there we go. It's pulled it in. Okay, so everyone feel free to play around with that. And the last thing I'm going to talk about before going on to the next exercise is the concept of shared data. So on Galaxy, the concept of histories, there's information that we want to make public to users. So input data sets that you can use for the tools, such as the MDP and the XCMS, reference files, reference libraries, anything that you can use to get started without needing to provide data yourself, things that can help give you a foothold into using the tool. So if we follow along here, I'm going to go back to Galaxy. So here, I think this is shared data. Right.
Did everyone get some green outputs? All their outputs are green.
Yeah, did anyone manage to
You've
upload?
managed to import some data ready to be used? Awesome.
One of these says histories or published histories. Is there some of these shared data? Does one of these says shared data?
This is the shared data.
Okay, one of them say histories. Yeah. Okay. Fantastic. Thank you very much. Okay, sorry about that. We figured it out. As well as reference input data files, we also make public histories. So this is a way for you to see the execution of a workflow like the one that Tom was showing earlier from start to finish, all of the inputs, all of the executions, and all of the outputs without needing to do it yourself. This is a way for you to be able to interrogate end-to-end the workflow without needing to understand completely what goes in and what comes out. So I invite you to click on this Shared Data tab, and it's this one here, history there, the second one down. I'm reasonably sure, history, yeah. There's the second one down and the third button across. Just as an example, I'll click into one and show you. So this is a metabolite study 2295 using the positive polarity that we've used in one of our workflows. I'll give it a second to load. So this is an end-to-end run of a workflow, retrieving data, pre-processing it, and then annotating it. You don't need to understand everything you're looking at just here. This is just to introduce you to the concept of a history, the inputs, the processes, and the outputs. If you click the View button here, you can interrogate the individual steps or the individual outputs a bit further. You don't need to, like I say, you don't need to completely understand what you're looking at the moment. Just to understand that this is a sequence of inputs, pre-processing, processing, and outputs. And Galaxy allows you to persist this exact sequence of events so that you can go back and peruse it later, but you can share it, people can reproduce it, people can use it as reference, however they see fit. So there's also reference files, so I'm reasonably certain what this says at the top is data libraries. You can see the stock data that we offer for certain tools, and we have reference files as well. I'm not sure which is which. So I invite you all to explore briefly, and then once everyone's had a chance to familiarize themselves with Galaxy MetaboLights Labs, we can move on to the next exercise. If anyone is having trouble with any of this, or they're a bit confused, please feel free to ask myself or Tom. We can do our best to make anything clear or to help you with whatever the issue is.
Yeah, anyone stuck so far? You now are all Galaxy experts. You'll be able to go and run any analysis in Galaxy. Okay. Just to say, there are great training material as well. If you go to any of the Galaxy instances, you'll see material there that will get you introduction to Galaxy as well. It's used, through all the different bioinformatic domains, from single-cell stuff to proteomics to metabolomics.
Okay. Okay. Feel free to play around with MetaboLights Labs for the next couple of minutes. If anyone has any questions specifically about MetaboLights Labs or Galaxy or our workflows, please feel free. And in a minute, we'll move on to the next exercise.
Everyone's happy? You're all happy? Good, good, good.
Okay. If everyone's okay with Galaxy and all of that's clear enough, I can move on to the next exercise. So this next exercise is twofold. So we want to get you on MetaboLights itself, interrogating an actual study, coming to understand some of the basic metadata about a study, some of the things that describe a study and its design, and we're going to have you execute a tool to convert some raw files from that study into an open source standard format that you can then go away and use theoretically for any tool that took that format. Okay, so first things first, if we navigate to this particular tool on Galaxy, so if you go to Tools, Get Data Metabolomics, and the MS Data Provider tool from MetaboLights database, I'll leave that information up there just for a minute, and then I will go on to Galaxy myself. Okay. Okay, so Get Data Metabolomics and MS Data Provider from MetaboLights database. Okay, so hopefully you've all got something like this on your screen. If you're still finding it, I'll give you 30 seconds or so. Okay, I'm going to move on. If anyone's having any trouble doing any of that, please let Tom and myself know. Okay, so this is where the second part of this exercise will also begin. So on the study selection input field of the MDP tool, you want to select MTBLS2469. But we also ask you to scan this QR code. This will take you to a Slido quiz. We also want you to go to MetaboLights itself and search up this study, MTBLS2469. We want you to bring up the study page. The poll questions can be answered by just clicking through a MetaboLights study, clicking into each of the tabs, and finding out some basic information about this study, MTBLS2469. So I'll give everyone a few minutes to do that, and I will show both selecting a study on Galaxy and navigating to MetaboLights, just for clarity.
Everyone happy? You have 2 minutes to answer the first question.
Okay. So just in here, you can type MTBLS2469, and it will auto-populate it for you. But we also want you to bring up on MetaboLights, ebi.ac.uk/MetaboLights2469. And then on the Slido poll, we want you to interrogate the study and answer the questions.
What was the question, Cal? You navigate to
Yeah, I'll put the Slido poll up. Next one. Sorry?
the first one.
This one? Okay, so the first question is, which type of analyses can be found in MTBLS2469? I'll give you another 60 seconds or so to answer this question.
Multiple choice, pick the right answer.
Sorry, I'll bring that back. I'll bring the QR code back up.
Everyone else happy? Yeah, we're good. We're good.
Just give people another 30 seconds or so just to answer this first question. Okay, I'm going to move on to the next question. Okay, so which journal was the publication cited in MTBLS2469 published? So again, this is a bit of information that you'll be able to get from the study page for MTBLS 2469.
Everyone's found the study page on MetaboLights, www.ebi.ac.uk/metabolites/the accession MTBLS.
Okay, I'll give everyone another 30 seconds or so to answer the question, and then I'll show you on MetaboLights how you can find out what the correct answers were.
There's no questions, you guys? No questions? Is all clear?
Okay, so I'm going to close the poll now, and then I'll show you how I can get the correct answer. Okay, so the first question was, which type of analyses can be found in MTBLS 2469? So that's a question about sort of the design of the study. So if you're on the MTBLS 2469 study page, you can navigate to the descriptors tab to find out some of the basic design descriptor information. And in the keyword section here, you can see untargeted metabolites. So the type of analyses that can be found in MTBLS 2469 were untargeted metabolites, and you can find that in the descriptors tab. Okay, and the next question, which was which journal that's cited the MTBLS 2469, which was where was it published? In this publications box here, you can just click the link, and it'll tell you. It should take you to a link here, and it was the Journal of Proteome Research.
Did everyone get the correct answers? Everyone understand? Good, good.
Okay, so I'm going to move on to the next step of the exercise. Okay, so on the Metabolites Labs tool, can you make sure that everyone has selected MTBLS 2469 as a study, because that's important for the next steps. Okay, assuming everyone has, I will bring up the next slide of the exercise. So this is, we need you to select a particular study assay. So there is actually only one assay for this study, and it's been auto-populated. But we want you to go on to this particular assay on the Metabolites study page and interrogate it. So if you scan this QR code, this is a new set of questions. The questions for this poll will pertain to this particular assay and information about the assay. So if we navigate to Metabolites, if you click in Assays, it's the first and only assay sheet there. And if you then go into the Slido poll, you can find the questions that can be answered by interrogating the assay sheet. So if I go to the next one, next Slido poll, Okay, so the first question is pertaining to this assay sheet. Which scan master charge range was employed for the LCMS assay in MTBLS 2469?
So again, 2 minutes to answer the question, find the answer on metabolites. I'll just explain a little bit about the assay concept. So one study can have multiple different types of analysis, right? You can think of it as simple as, I want one assay for positive mode, I want one assay for negative mode, I want one assay for GC, I want one assay for LC, I want one assay for NMR. And MetaboLights has worked with the community to have templates for these assay types. Again, so all the metadata is there, easy to query, linked to ontologies. Yeah, so any questions, let us know.
Okay, I'll give everyone another minute or so to go through the assay sheet and try and find the correct answer to the question about the master charge scan range. Okay, I'm going to move on to the next question. So the next question is, what was the accession for samples from humans, Homo sapiens in MTBLS 2469? So we're looking for a particular ontology ID. So this is information that you will only be able to obtain if you actually download the sample sheet. So if you go into the files section here, and you click the little blue button here next to S_MTBLS 2469. If you click download there, it'll pull it in, and then you will be able to go into the sample sheet and you will find the entry for Homo sapiens in the sample sheet, and it will give you the correct ontology term. So I will go back to the Slido poll, give everyone 2 minutes to download that sample sheet and find the correct ontology accession. If anyone's having problems downloading MetaboLights data or the sample sheet doesn't look right, please let me or Tom know. We can come over and try and help.
Yeah, so I should probably introduce you to one more concept here. So MetaboLights has assays, but it also has samples. So I said you can have multiple assays per study, but you can only have one samples per study. And this is because you might have multiple assays, but they probably analyze in the same sample. So for positive and negative mode, I want to keep that relationship, that sample 1 was analyzed in positive mode in assay 1, and sample 1 was also analyzed in positive mode in sample 2. So for this, when you're talking of how can I get sample descriptions, I have to go to the sample sheet. Does that concept all make clear? And in the sample sheet, we ask for some really default characteristics. What organism were you studied? What organism type? And what organism part were you studying? Was there a variant associated to this organism? And also now we're starting to ask the community, what kind of sample type is this? Is this simply an experimental sample? Is this a pool QC? Is this internal standard mix? And then the other thing we encourage submitters to give us is also factor information. So this is really key on how you describe sample by sample, how they differ from one another. Are they different treatments? Are they different time points? Are they different diets? Are they different diseases? Again, all captured in the framework of good, fair data, make your data findable, accessible, interoperable, and reusable.
Okay, I'll give everyone another 30 seconds or so to go through the sample sheet, and then I'll move on. Okay, I'm going to stop the poll now, then I'll show you how to get the correct answers and what they were. Okay. So the first question was, what was the scan range for the assay, the LC-MS assay in MTBLS 2469? So if you scroll into the assay sheet here and you scroll along, you can see there the parameter value column, scan master charge range. It was between 100 and 700. So that's a basic bit of information about the LC-MS assay that you can find quite easily just browsing the assay sheet. So hope everyone got the right answer. So the next question was, what was the accession for samples from humans or Homo sapiens in MTBLS 2469? And for that, you needed to download the sample sheet. So I will bring it up myself. You can see here it's a text file. It looks a little bit raw, but you can see here, so characteristics, Homo sapiens, organism, and term source reference, which is NCBI taxon 9606. We don't render this information directly in the browser all the time, just to save on space and readability. But if you want to interrogate the information further, it's always best to download it. Okay, did all that make sense to everyone? If so, I can move on to the next part of the exercise.
Any questions? Everyone got to that stage?
Everyone okay? Cool. Okay, so the next stage is just selecting an assay description. So this is a collection of characteristics about the given assay that we filter raw files for the conversion process by. So polarity, column model, column type, organism, and part of that organism. So for this particular study, there is only one such set of characteristics, which is positive, reverse phase, that particular make and model of a column, and Homo sapiens with blood plasma type samples. So you should see that auto-populated for you in Galaxy here in the assay description. It is indeed the only one that you can select. So if for some reason that hasn't been selected for you, please let us know, because something has gone wrong. Okay, simple enough. Next stage, again, so the polarity filter. Another basic way of sorting through the files. There is also only one polarity option for this study, which is populated for you, so you don't need to do anything. And then the only spectral file format available on the study is RAW. And again, this has been selected for you. Other file types can be derived, but there aren't any derived files in this study. And for the conversion process, we want RAW files. Okay, so if that makes enough sense to everyone, we can move on to selecting the spectral files we want to convert. So we ask, for the sake of the last set of questions, that you use these exact file names for the conversion process. So I'll bring these back up in a second, but if I just show you, if you just scroll down, they'll be in this list, which I recognize is a little bit long, but they'll be here and here, and then again towards the bottom. So I'll bring up these slides just for 30 seconds or so, for everyone to tick each of these 4 input raw files for the conversion process.
Yeah. So here this one study only has one assay and one type of raw data files. If you had multiple ones, the drop-down menus would show you also the multiple ones. So if you had positive and negative 2 assay sheets, you could see in the drop-down menu that there'll be 2 assay sheets for you to use.
Okay. If anyone hasn't done that yet or is having a problem finding those files, please let us know. But for the time being, I'll move on.
And then just a reminder what this tool will do when you click the execute button. So here you now selected, I want to use this public study. I want to use this assay. You've told it what kind of data you have. I have positive mode data. You've told it what kind of data files you have. You have .raw data files. And you've told it then which data files you want to start to process. Here we're only choosing 4. We're only going to choose, just 2 QCs from the start, 2 QCs from the end, primarily so you don't overwhelm our server too much for this tutorial. But in the real world, you can choose the whole assay if you want to run all data files to pre-process.
Okay, yeah, so there's a QR code for the last set of questions. And I'll leave the QR code up there for just a second. But then once you've got the QR code ready, you can click execute. So I'll do the same. I will select those same files. So, okay, and you can click execute at the bottom. So once you've done that, you'll be given a summary page telling that you've executed this particular tool with these inputs and it will produce these particular outputs. You'll notice to the right in the history column that your job will either be grey or orange or green. So grey, that means it's just been queued. and that it's waiting for a node to become free to execute it. Yellow, as you can see, means it's running, it's doing its thing, and when it eventually turns green, that means it has successfully converted all of your files. So while people are waiting, while people's jobs are queued up and executing, I will go back to this page just for a second, just in case people didn't get the QR code. I'll leave that up for another 30 seconds or so. But this is for the last set of questions, and they will pertain to the files that you've just converted and viewing them, interrogating them on Galaxy.
Yeah, we want to see grey, orange, or green, which means your job is pending, it's being processed, or it's executed correctly. Let us know if you see any reds. Hopefully not.
Yeah, red is bad. Let us know about red. Okay, I'm going to navigate back to Galaxy. No, I'll navigate to the Slido poll, just for everyone to have a look at on the big screen. So it's this one. Okay, so I will show you exactly where to find the answers to these questions in just a second. I'll give everyone 30 seconds or so, just in case they are still waiting for their jobs to execute. But if you navigate back to Galaxy, you can see mine are still running. So I'll wait for my own jobs to finish so that I can show you how to interrogate the results. If anyone's having a problem executing, if anyone's jobs have crashed or errored, please let me or Tom know. We can come and see why. As soon as mine finish, I will come and I will then show you how to get the results that you need to answer the questions in the Slido poll. Sorry, Tom, you go ahead.
Everyone should have 5 objects as their results that we need to explore and understand. Okay. Everyone executed the tool?
So this is a Galaxy instance for purposes like this. So sometimes when you concurrently execute a load of jobs, this can happen. They can take a while to run. So.
Has everyone got green? Hands up, who's got green? Oh, we got a few greens, that's good. Everyone else is
Everyone
orange?
else is executing. Okay. That's a little slow. So I'll give it another 30 seconds or so, but if these jobs stay running for a bit too much longer, what we can do is we've prepared a history that shows the outputs from this exact execution with that exact study, those settings, and those raw files, so we can all, on your machines as well, import the history and get the answers without needing to waiting for these to run. So I'll show you how to do that now. So mine have just finished executing, obviously. Has anyone else got green? Has anyone else finished executing? Okay, this is easy. If anyone hasn't finished executing but you're waiting, can you put your hand up? Okay, a handful. Well, I can just show you how to get the answers to the Slido poll in just a second. I'll put the question back up on the board. Hopefully you've all got it on your machines. So what was the lowest observed master charge in the first spectra on this particular file? 02.12202, QC pool set 1 underscore P underscore 2. just share them out. Okay, so if you go into the Shared Data tab at the top, the third one across, and you go into the Histories button there, the one we looked at earlier, you can view, this is just in case your job has not executed successfully or it's still waiting. Just in the interest of time, we can view one that we did earlier that has the exact same inputs and outputs. So it's the one at the top there, METSOC 2024 Exercise 2. So if anyone can't see that, please let us know, and we can see why.
Yeah, hopefully everyone else is running on their local machine. But if, in case you don't, we have a backup. You can go to the shared history, and you can then start to interrogate these results while you wait for your jobs to be executed.
Okay, so yeah, shared data, histories, and the one at the top, METSOC 2024. Okay, so the first question was, what was the lowest observed master charge in the first spectra in, for God's sake. What was the lowest observed master charge in the first spectra in 022102 set 1P2? So to get that information, you can click in here, and you can find that particular MZML file. And it will load it in here. And if you scroll down to the lowest, if you can search for lowest observed master charge, you can see on a tag down here is the particular tag. Okay, so I'll give everyone 30 seconds or so to open that particular file from Galaxy. So again, you go in here. If you don't have it yourself in the History tab, you can go in your own history. You can use the shared data. Click into the list of MZML files, open that particular file, 022102, and find the lowest observed master charge. If anyone's having any trouble with that, or they can't find a particular file, please let us know. Okay, I'll give everyone 10 or 20 more seconds, and then we'll move on to one last question.
So here, remember, you're taking the raw data files, you're creating an open standard, the MZML format, which you heard from the GNPS guys earlier, and now I'm interrogating these MZML files. Any questions? You guys have 10 minutes left, and you've asked no questions. Everyone can reuse public data? Is that what you're telling me, experts?
Okay, we can move on to the next question. Okay, so in your history or in the shared history, you will have also been given an applied configuration file. So in that applied configuration file, what is the first filter used for the MS Convert process? So if you go into Galaxy here, you can see the applied configuration file. If you click View there, it will tell you, or even in the drop-down. And there, you'll be able to get the correct result.
Yeah, so I should probably share with you how you create MZML files, right, from your raw data. So there's a very popular tool, a very good tool called MS Convert from Proteo Wizard guys. So yeah, you can download it, install it locally, you can run it on your machine, on your data, but we put it in the Galaxy so that we can use it on public data that will then produce the open standard that you can then start to plug into all the different other pre-processing workflows you guys want to use, whether this is MS1 level simply for peak picking and XEMS, or whether this is MS2 level to go off to GNPS and do your molecular networks.
Okay, so in the interest of time, I'll leave this last poll question open for the next 30 seconds or so, and then I'll explain how to find the answer to those two questions, and then if there are any questions at the end, we can field those. Okay, so I'm going to stop the poll now. Okay, so the first question, which was to interrogate this particular output MZML file. If you viewed it in the browser and you went down to lowest observed master charge, you can see the value was 100.04, give or take. And for the second question, what was the first applied filter for the configuration file? You can see here the filter was peak picking vendor mass spec level is 1. Okay. So for the next, that's the end of the exercise, but I will move on just to end with some last slides about multi-omics.
I put in the description about exploiting metadata and multi-omics, so I thought I better not disappoint.
Okay, so we'll save the questions for the end, but we'll quickly go through the multi-omics stuff just a second. Okay.
Yeah, so we're excited about public metabolomics data, and with the GNPS guys, we're super excited. But what we're also excited about at MetaboLights is multi-omics data. And this is really one of the key benefits of EBI, because at EBI we have all the repositories. We have ENA, the European Nucleotide Archive, for your genomics data. We have PRIDE for your proteomics data. And we have Magnify for your metagenomics data. Bioimage for your image archive. Now we're starting to see these big projects roll in. We're starting to see COVID Data Portal have multi-omics data. We're starting to see a big EU project, Hollow Food Data, having multi-omics data. And also Trek have multi-omics data. So I take this one sample and I do all the kind of analysis that you imagine. So I want to start to link on the sample level. So at EBI and MetaboLights, I'm particularly excited about this. We have one example now called Hollow Foods. So now we are linking samples. All data types at the sample level for you guys to use. So there's the Hollow Food Data Portal. You can go to there, you can say, at this sample, go give me the metagenomics, go give me the genomics, or go give me the metabolomics, bring it into the environment so then I can start to reuse this public data. So yeah, go check out the data portal. They're linked with Jupyter Notebooks, R, Python for you guys to go and use. And again, we want more of this. This is really valuable public data that you can reuse. You can start to build new insights, multi-omics insights. You can start to develop new tools around this area as well. So come check MetaboLights out for the multi-omics. And this is just a list for you guys of your awareness of all the resources at EBI. From chemical molecules and drug discovery, to genes and genomes and RNA, proteins, imaging and cellular structure, genetic variation, disease data, and literature, knowledge and management. Yeah, this is a team. We're only a small little team. 4 of us are here today. So come find us through the conference, and we'll be happy to chat. Yeah, we're welcome to feedback, so let us know. And yeah, hopefully you guys have enjoyed the workshop. We'll all be around for the conference, so come and catch us. And hopefully we made it a little bit easier for you guys to start to reuse public data and to get excited about it. Thanks.
If I can say one last thing, since you have attended the workshop, we have presents for you. We have GNPS stickers. So if you want one, I'll just pass by and I'll give it to you if you want them.
And also we're all at the conference, so whenever you have more questions, you can always approach us and yeah, just ask away.